Ensuring the safety of medicine is pivotal for pharmaceutical industries, and safety concerns identified during the almost 15 years of drug development can often lead to project delays and late-stage drug attrition. Therefore, designing out safety liabilities at a very early stage in the drug pipeline will help to bring safer medicine to the market, as well as save time and money for pharma industries. But it's also important to characterize the adverse effect to avoid dreadful events after the launch of the novel drug on the market. But a question still remains: how we could improve the predictivity of adverse effect before the novel drug is launched on the market? Hi, I'm Martina Cherubin, early stage researcher number 2 of INSPIRE, which is a European training network in the field of safety pharmacology, and I'm based at UCB Pharma in Belgium. Today, I'm going to illustrate you my PhD project, which aims to develop and validate a cardiomyocyte assay to better predict cardiac liabilities that influence both function and structure of the heart. In other words, my project will be focused on in vitro-based assay of human-induced pluripotent stem cells derived cardiomyocyte, which resemble quite reliably heart cells. The ultimate goal is to foresee potential adverse effects of novel compounds and ensure the safety of the final product before it reaches clinical phases and the market, and finally, reach the patient. To go into further details, the first part of the project is focused on the identification of an in vitro assay based on human-induced pluripotent stem cells derived cardiomyocyte that could detect the effect of several compounds and drugs that are already present on the market, based on the recording of several parameters, like, for example, viability, contractility, and electrophysiology. The second part of my project is focused on the identification of novel biomarkers of cardiotoxicity, in particular with a focus on microRNAs. MicroRNAs are small non-coding RNAs involved in several cellular processes by modulating gene expression at post-transcriptional level. They have been found associated to several phases of cardiac development, as well as in the pathophysiology of several diseases. Due to their characteristic, like, for example, stability in biofluids, high expression after a substantial damage, and tissue specificity, they could be considered as promising biomarkers to predict cardiotoxicity. The third and last part of my project is focused on the assessment of translatability of previously generated data in iPSC-derived cardiomyocyte to, first, in vivo models of mice treated with doxorubicin, which is an oncological treatment known for its important cardiovascular adverse effect. For this study, we collect the heart of the mice as well as the blood and the plasma, and we are going to investigate biomarkers. The last part of my project will be to investigate this translatability and the assessment of this translatability to patient-derived cardiomyocyte. We're going to collect blood from different patients in collaboration with University of Maastricht and Antwerp to generate stem cells from the patient.